Our previous work financed by Novalait have allowed us to understand that the farm management practices at the farm have an impact on the microbiological quality of milk. However, we make as a hypothesis that modification, the microbiota of the milk is governed by biofilms that develop in milking systems at the farm or in the dairy equipment of cheese factories. Thanks to the Coalition of Research for Optimal Quality Milk, Opt plus Lait, there is a multidisciplinary project. We're bringing together researchers from Quebec and Ontario, present in 3 different universities, and 1 research center on agri-food has been able to appear. The objective of this program is to compare microbial communities involved in the production of biofilms. To really characterize the microbiological films microbiologically, first of all, you have to sample those films. So far, 5 farms with conventional milking systems and 6 farms with robotic milking systems have been sampled. During those samplings, we use swabs, sterile swabs like these, which are then dipped so as to harvest bacteria that adhere to the equipment despite the cleaning of the milking systems. The sample is then ready to be conserved and analyzed and sent to the lab to be processed. The microbial count will be done, as well as the identification of bacteria by sequencing, high flow sequencing. The objective of my master's project is to evaluate the presence in starters of food bacteria, Campylobacter, which is strongly prevalent in dairy cows. To do that, after taking samples at the farm, they put in a culture to allow Campylobacter to multiply. After enriching, the sample is put on an agar gel like this for a molecular identification. My project allows us to see if the biofilms are threatening the quality of milk by taking part in the propagation of pathogenic bacteria such as Campylobacter genu. To understand the impact of the microorganisms on the formation of peptides, as well as the various parameters governing their production, we use a bioreactor. This is a method of study that is dynamic to create shearing forces and to reproduce conditions close to those that you find during production and milk processing. We adapted the protocol of the STM to be more representative of the actual conditions at the farm. Actually, microfiltered milk will enter in this reactor where it will be in contact with the biofilms that have been previously sampled and cultivated in bacteria that are problematic for the dairy industry. Then it is recovered and preserved at 30 degrees for a duration of 24 hours to check whether the biofilms are contaminated. The knowledge acquired during that project allows us to target new biofilm control strategies, such as testing the effectiveness of those bioprotectors on models that have been developed. As part of this project, we're interested in identifying bioprotecting cultures and identification of their antimicrobial properties. Lactic bacteria produce several antimicrobial components, especially bactericides and other lactic compounds. These compounds are very promising alternatives to the disinfecting agents in the food sector. As you can see on these GLOs, we were able to identify a bioprotecting starter that can inhibit the growth of a targeted bacteria and also a mold. We also work on evaluating the inhibiting potential for inhibiting the dairy films produced by lactic bacteria that are thermoresistant and which present a major problem for the dairy industry. The anti-biofilm effect is studied using a test on a microplaque like this one. At the end of this project, pertinent information on the impact of farm management practices or in the field on the appearance of biofilms will be generated.
From cheese manufacturing allows us to look at the microbial aspects associated with products, including cheese.